So first of all, thank you very much for giving me this opportunity. It's really a pleasure. My first time in Grand Rapids, and I've been having a wonderful time at the conference so far. And it's a particular pleasure to speak in front of all of you and people with Parkinson's and their caregivers and loved ones. So it's really a pleasure. And so what I will be talking about is actually a little different from what you heard today, earlier today, from Dr. Bjorklund about using stem cells to transplant and treat people. What I'm going to talk about is how this new technology of deriving stem cells from patients will actually, I think, help us understand the disease better to come up with new treatments and possibly cures. And I'm going to try to convince you why that's the case. And I will also say that I'm going to try to gear this to make it as simple and non-technical as possible. But I do want to encourage you, if I'm getting too technical or if things are not clear, please stop me, raise your hand, and I'll be happy to explain things a little more clearly. So here's, again, my lab has been interested for 2 decades now in trying to understand what the causes are of Parkinson's disease and the real deep mechanisms, because I feel like that's the only way we're ever going to get to a cure. When I was training as a neurologist in 1995, a long time ago now, almost 30 years ago, these were what were described as the causes of Parkinson's disease. So the loss of dopamine, maybe something in the environment like smoking or infection or rural living like pesticides and well water and toxins. And really I was taught that genetics is not important for Parkinson's. And now we're going to fast forward to now, and you see that there's now a whole slurry of genes that in some cases cause Parkinson's in a familial form, but in other cases are just risk genes. And in fact, in just a few years ago, and you'll be hearing more about this in the main sessions, there are these studies called GWASs, or genome-wide association studies, where now there are not only these genes that cause familial Parkinson's inherited, in clearly families, to risk genes that if you have certain varieties of these different genes, and there's almost 80 of them, then that increases or decreases your risk of Parkinson's disease. So you see, we've gone from a disease that was thought to be not genetic at all to now a disease that has like a plethora of different genetic components. And I think this is important because if you're going to get to the bottom of it, you want to understand the cause. And so my lab has been interested in working on several of these genes. But how we work on these genes is important. And you heard from some of the talks today about ways that people work on them. And they're all very important, and they've led to important advances. For example, mice or rats, or using cells that are grown in the lab, and how that can help us understand the disease. But in fact, if you really wanted to understand the disease, you'd want to get the kinds of cells that are actually important for the disease. And that would be human cells, human neurons, or human brain cells. Now, most people, even when they have Parkinson's, they like to keep their own brain cells. They don't want to give them away. So that's been impossible until recently, to actually study people with the disease to study their brain cells. And I want to emphasize how important this can be. If you think about diseases like cancer, almost every person with a cancer will get their cancer resected or a biopsy. The cells from that biopsy can then be used by scientists to understand the disease. And that's led to incredible breakthroughs. Some people with cancers now don't even get chemo anymore. They get targeted treatments because the cells from their cancer can be studied by scientists, and you can figure out what's wrong with those cells. In Parkinson's or other brain diseases, it's very rare you get a sample of someone's brain. So you couldn't do that until recently. And this is where the breakthrough has come over the last 10 or 12 years or so, where it's become now possible, thanks to just even a simple blood sample, sometimes a urine sample, to collect cells that aren't brain cells, turn them into induced pluripotent stem cells. These are cells that they're stem cells, but they're induced from somatic cells, from cells like from skin or blood. And once they're a stem cell, you can turn them into any kind of cell you want. And in our case, we'd like to turn them in, for example, to dopamine neurons. So this has now become possible. But the question is, and we've been talking about this now, as I said, for more than 10 years. So what's come of this? I mean, if you really want to understand why I'm passionate about this, this slide will more or less, sum it up. This is how we discover drugs in general. You think of a protein target, so something that is made by a cell, a protein, and you develop, so that target, you think if you change that target, if you have a drug that binds to that target in your cells, it might help the disease. So you then screen for compounds, you take that drug, prodrug, and you inject it into animals, and you have an animal model of the disease, a mouse model, for example, and the drug helps or cures the mouse. So then you take that drug and you do clinical trials. And I'm sure many of you have either been in trials or have heard about trials. And then you look for it to work. And we failed miserably using this technology, at least for Parkinson's and even for Alzheimer's, to be honest, we haven't really been very successful. So here's the idea behind why stem cells, I think, can change all of this. This is what you can do with stem cells. Each of the people with the disease, you can take some blood, turn their blood into stem cells, and then grow the kinds of neurons you want. And then what you can do is you can take this approach, and now instead of doing the screen where one size fits all, one drug fits all, you can look if each individual person's cell will respond to a drug. You can skip this part with the animal, and you would only do the trial in the patients whose cells responded to the drug. So this is a kind of personalized therapy. So this is what the holy grail, I think, is to, instead of saying, like, if we give the drug to everyone, apparently it'll fail, but maybe it would have been successful if you took the 4 or 5 people here that might have been sensitive that you could not know about unless you use this approach. So this is what we've been trying to do at my institution over the last, I would say, 6 or 7 years. And this has been a long-standing now collaboration with Thomas Durkin, who's a colleague there, where we've built, because our institution has a hospital and a clinic, we get patients to consent. They give some blood samples or skin samples or urine samples. We collect these in our biobank called CBIG for Clinical Biological Imaging and Genetic Repository. Tom and his team then reprograms these cells into stem cells, into iPSCs, and then we could actually modify these cells so that they're, if they have a mutation in one of the Parkinson's genes, that could be corrected. And then we develop these assays and we do screening. And doing this now, we've developed lines both in-house and through external cell lines. We have, over 100 lines from patients with Parkinson's, some with mutations, some without. And we've also engineered through genome editing mutations into these lines. And that allows us now to make different kinds of brain cells, either dopamine neurons, or as you heard about some of you this morning, glial cells like microglia. And so this is one example where we take these cells and then we differentiate them using a number of growth factors and differentiation factors. And then we can measure, for example, how they express different markers that would be normally expressed in dopamine neurons that are in our brains, like tyrosine hydroxylase, for example. And you can see that as these cells mature from 2 to 4 weeks that you grow them, they get almost 80% of them express this enzyme. So this has become a very useful tool in our system. And then you can develop assays to test, for example, and I'm not sure this is coming through very clearly, but here we have a cell line from a patient with a synuclein. Alpha-synuclein is one of those Parkinson's genes that is the first gene that was discovered. And so this person has extra copies of synuclein, making him synuclein levels high. And therefore, if you put fibrils of synuclein, you get this pathological signal that you can see here. and you can genome edit and correct the line where now if you put the same amount of fibrils, you have much less pathology. So this allows us now to use this system to find maybe drugs and see if this patient, if you do this now with a drug, you can reduce this back from this high level to this low level. And then that drug eventually can be brought into the clinic. So let me just stop here for a second. If there's any questions at this point, I mean, I'm happy to take them. Yes?
Does your procedure,
Can anybody use this, or is it just limited to your lab, or is it
No,
public
this is
knowledge?
no, this is something that's available in many, not everywhere, but in many centers. I know here at the Van Andel, they also have an iPSC platform. So it's possible to do this in many places. And these are the same types of the blood draw, turning them into the stem cells are the same stem cells that we heard about earlier this morning, that people are using these stem cells for transplantation. I'm just, my own bias in a way is to think that the bang for the buck, I think, would be higher to try to find a new treatment using these patient cells than replacing cells that are from someone who has Parkinson's, just because, you know, we already have good therapies like, you know, even Cinemed and levodopa are already there to replace the dopamine. What we want to do is not so much replace the dopamine, but rather cure and slow and stop the disease. So that's sort of the angle. But yes, it's widely available. Yes.
I feel like I misheard something you said about alpha-synuclein and being a gene. And then I just Googled it, but can you just clarify that?
Sure. Let me quickly go back to this slide. Yeah, so these are some of the genes in the last 20 years that have been discovered. Up here, this first gene, in 1997, called alpha-synuclein. It has another name called SNCA. And so some people, in rare cases, some people in families have mutations in this gene. And those people get Parkinson's disease with high likelihood.
Why did I think that alpha-synuclein was a misfolded protein?
It is so the gene encodes a protein. So all of these genes, typically what happens is the gene is like the blueprint, and then in the cell, the gene leads to transcription of another chemical called RNA, and that RNA is translated into a protein, and then that protein misfolds. So the reason mutations in this gene we think lead to Parkinson's is because the mutations increase its likelihood to misfold. And so alpha-synuclein, you're absolutely right, normally shouldn't misfold, but when there's too much of it or when there's a mutation, it misfolds more than it should.
Is that is PARK1 the only gene linked to that folding?
That's a complicated question. There are a lot of genes. So we think in Parkinson's disease, alpha-synuclein is the main protein that misfolds. But a lot of the other genes here that have mutations can also influence how much alpha-synuclein misfolds. For example, GBA1, which was discovered a few years later, even though itself probably doesn't misfold, or if it does, what its main effect in Parkinson's is that when it misfunctions, it leads to more synuclein misfolding. So these genes, some of them have interactions that are sort of complicated.
Is there is the misfolding of the alpha-synuclein, well, it's kind of like a two-parter. So is the misfolding of the alpha-synuclein, does it always lead to Parkinson's? Or can you have alpha-synuclein and never have that expression? And the second part of that is, can you explain tau in regards to that?
So that's a complicated question. I actually don't know the answer. I mean, I think typically if you have a mutation or too much synuclein, typically will end up misfolding over the course of a lifetime, and it will greatly increase your chances of getting Parkinson's.
Can I just add one quick thing?
Yeah.
Everyone has alpha-synuclein, right? Okay, so normal basal levels, unmutated, everybody has it. It's just the misfolding that we think is contributing to disease. Just one point of clarification.
That's a really important point, actually. And the idea is that you can imagine these things are not as simple as they may seem, because as you're I think implying, we have billions of copies of this protein once in all our cells over the course of decades, and you only get the disease 50 years later. So it's all a question of risk. You're shifting the balance for more of a risk. Probably it may aggregate once in a while and then get degraded, but when it's overwhelming the system, it aggregates too much. And there's another question.
So I've had induced pluripotent stem cells done 4 times with 4 different groups. And I have the LAR2 G2019S variant. But I had it done, I'm a non-manifesting carrier, so I still do not have Parkinson's. So my stem cells that were taken or created 10 years ago, the first time I had it done, are they going to look different than the stem cells that I had done just recently, like a couple of weeks ago at the NIH? Or is this something that's looked at longitudinally to say, do they look different now than they were?
That's another amazing question. I mean, I struggle to answer that question for a couple of reasons. And I'm sorry, because we might be getting a little too technical, but one thing is that the stem cells are able to generally faithfully capture your genetic makeup. So let's say if you, as you said, you have a LRRK2 mutation, that's going to be kept, with great fidelity, whether you have it today or 10 years from now or 10 years ago. What it doesn't capture is all the epigenetics. So let me give you a crazy example. Like if you had a really severe infection or you were exposed to pesticides in the interval, then that may affect your health in general, but probably at the step of reprogramming the cells into iPSCs, that may be reset. So this is a great model if you're interested in capturing the genetic components, but there's a whole other world of epigenetics, like as I said, that you may lose along this. Those are amazing questions. I'm really quite impressed with that. And I mean, we may not get to the end of this talk, but I think it's more fun to discuss these things than necessarily hearing me talk on my own. So let me just try to continue where we left off. Right, so this is just to give you like a bit of a background of an example of a phenotype we call, or what it looks like, why if you have this kind of mutation, which is a triplication, you have a higher propensity to get this kind of misfolding. And when you correct it, you can correct also the phenotype. And so that's what we're looking for. We're looking for establishing reliable models of cells of patients that we can then screen with thousands and thousands of different possible drugs to see if any of them will reverse it from this back to this. And then we can go back to that patient and do a trial in that patient and a subset of other patients who behave the same way. Now, here's something else to make it maybe even more complicated. And a little bit more complicated, but I think this is super important. And it's more about the sociology of science than really actual science. So this is, the human genome was sequenced in 2000. So it's been 25 years since we know the sequence of all the genes in the human genome, all 20,000 genes. And this is, so here you have all from 0 to 20,000. On this axis, what you're seeing is publications. So you'd think that after 25 years, scientists would study all the genes pretty much the same. I mean, there's no reason to think this gene is more important than this gene. And yet some of these genes, so here's this one, it's just called P53. It's a very popular gene, obviously. But then there's like over 600 genes with 0 publications, even though it's been 25 years, okay? And that, you may say, well, maybe these are all the disease genes, so everyone wants to study these. No, if you look at GWAS, remember that genome-wide association study, not just for Parkinson's, now we're talking about all diseases, you see that there's a little more here, but there's quite a few there. And if you think monogenic diseases, well, they're throughout. So what we're going to say, we're going to call this the dark genome that nobody studies. And if you look at Parkinson's disease, and the reason I tell you about this is because Parkinson's, we're not better than anyone else. It's exactly the same. So this paper came out 5 years ago showing these 80 genes, new genes that are risk genes for Parkinson's. Now let's do the same exercise in Parkinson's. It's the same thing. Everyone's studying SNCA, which is synuclein, and then a few other genes. And then there are all these other genes that no one studies. So one thing that we're starting to do is trying to come up with a way where we could systematically, because there's, if you go back to this, there's maybe the cure for Parkinson's will be when we manage to unaggregate or misfold synuclein. But it might also be that a drug against gene number, 37 here that no one knows about, if there's a drug for that, maybe that'll regulate all the other stuff and could still be a very good cure. So we're a project now that was started a few years ago, a pilot project funded by the Fox Foundation and the Structural Genomics Consortium, which is a Canadian organization for open science and drug discovery. We set out to try to prioritize these genes, these 80 genes, depending on a number of criteria, and then use these stem cells to now, all in the same stem cell background, individually knock out, using the technology called CRISPR or gene editing, knock out these genes one by one and see what, and do this, and then turn them into neurons, dopamine neurons, or different kinds of glia. And then look at the assays, like the assay I just showed you with the synuclein misfolding, different assays that we think are sort of typical or canonical Parkinson's assays. And then also look at unbiased assays like omics assays, like looking if there are anything that are shared by these different genes and see if they can converge into pathways, and then try to understand this at a more systems level in order to eventually come up with new prodrugs or antibodies that could modify these targets. So this project now, and here's an example of just one of these genome-wide association studies, one site in the genome. You can see these are all the different markers. And even in one of these 80 sites, there's more, these are all the genes. You can see there's multiple genes. So we had to find a way just by bioinformatics to decide which gene was the important gene for Parkinson's disease. So here with a colleague of mine, Ziv Ganor, who's a geneticist, we just published this paper ranking those, the genes in those different loci, in the 80 loci, into the likelihood that the gene is the gene important. And here, for example, it's this gene CLCN3, and I'll come back to it later. And then ranking them based on a number of things, including their darkness, in quotation marks, meaning that we didn't want to work on things that everyone was working on, like synuclein. We wanted to work on the ones on the right side of the graph that nobody was working on, to try to find new biological information. And just out of this purely kind of informatics data, we identified, for example, this pathway in a lipid phosphate pathway called inositol phosphate, where 4 of the genes function together in a pathway. And really, there's almost no papers looking at these 4 genes in Parkinson's. And yet the evidence becomes quite strong when you think that there's already 4 genes out of these 80 that are linked together. And so we picked a number of these, and just to give you sort of the red boxes, and then we we made the knockouts, and we used the more traditional genes as controls, and then we made dopamine neurons, and then we did Parkinson's targeted assays like the ones I showed you earlier. For example, the synuclein aggregation, mitochondrial function, etc. And then some of these omics. And here's just an example of like knocking out multiple genes here and looking at their effect on their expression of dopamine markers like TH, at their formation of connections or synapses, where you could see the different genes have effects in these pathways. Whether they're linked to their causality or relationship to Parkinson's, we still don't know. And then the synuclein assays, which I showed you earlier, and I'm going to go quickly here. And then just to give you one great example where this gene that I mentioned right at the top, CLCN3, we made a knockout of it, and you could see it forms less dopamine neurons than the other genes, and it also has, but it has relatively normal synapse number, but it has this dramatic effect on mitochondria. The mitochondrial membrane potential is abnormal. And then this pathway involving two other Parkinson's genes, PINK and Parkin, is affected in a very dramatic way. And you've got this box here, but you see this is this little red square here. It's much lower than the other genes involved in that we've knocked out. So just by doing this exercise, this gene that nobody works on, that is in a risk locus for Parkinson's disease, because we looked at these assays now, we may have implicated it in this pathway involved in mitochondrial quality control. So this is just to show you where we're going for this. And I think this is a bit technical, so maybe I'll skip over this. And maybe I'll stop right here because that's already a lot of information for one session.
Wow.
Yeah.
Thank you. I'm sure there might be more questions. Anyone questions? More questions?
Did insurance pay to do this?
Sorry,
Did insurance pay to do this?
I'm from Canada. But no, this is all research, so insurance doesn't pay even in Canada. It's all on research funding.
Okay, so
Yeah.
the patient doesn't pay.
The patient doesn't pay. No, you get research grants to do this, and this is something that is part of research projects at this point. Yes.
How do you mitigate against the mutations happening in stem cells when they, thank you. How do you mitigate against the mutations? Because my understanding is they live longer than other cells or could carry tumor risk from the donor or whatever. How do you mitigate those risks?
So for us, it's the problem is not as important, I think, are you alluding to when you're doing transplants, right? I mean, so that's not what we're doing here. As I said, this is for research to understand the biology, but we do all the cells when they're reprogrammed into stem cells undergo this very, very rigorous quality control. So you make sure there's genetic stability. The lines are sequenced. So if there are mutations that are incorporated, you see them. And they keep their stemness using a number of tests. So, you know, we try to be as rigorous as possible to make sure there are no mutations. But of course, over time, just like in our own bodies, you heard some talks this morning, we get somatic mutations, and it's possible that as the cells are grown up, there are mutations that develop over time. So you have to always go back to the source line and make sure you keep regrowing them. It's a little different, I think, with the transplantations. Again, that's not what we're doing here. There, you're not typically getting stem cells that are being transplanted. You're getting the differentiated neurons already. So those presumably have, you know, depending on the trial, have been tested.
And we've got a talk on that tomorrow morning.
Great.
Well, now I feel like I should just wait for the talk. But that was kind of my question I believe the subject of your talk was how this impacts us and, you know, the patients. And that's what I'm curious about is like timeline wise, how long, I feel like because you're doing it in, you know, lab created cells, like your progress will be much faster. But how do you choose which ones that you said it was kind of, you know, the one tied to alpha-synuclein, I have to assume is because you were seeing those plaques in your brain, so you reverse engineered that one back. But how long do you think it will take to get all the way through the ones that you mentioned?
Okay, here's my view. My view is that if you want to cure the disease, ultimately, you're going to have to figure out how the disease happens. And I think this is a tool to get there. I think it's a completely different question to take these kinds of cells and put them back in someone's brain. There, really, you're not, I mean, you're providing cells that make dopamine. So it's not the same question you're asking. So I don't think we can really talk about what's faster or slower. One, we're trying to understand and find new drugs, for example, that might affect this. And then hopefully we'll be able to give that to all of you who need them, which is a very different question than just taking these cells and putting them in. Because as you heard again this morning, even the cells you put in, ultimately the synuclein that's in the brain goes in and contaminates and crosses over into the transplant. So until you figure out how to stop that process, I mean, I'm not saying it's not going to be helpful in some way. I mean, maybe it will help some people, you know, go with less treatment, less side effects, possibly. But I think this is a very different question. So it's hard to compare them.